These are the lecture notes for Chapter 4 on cell structure and function. There are 8 sections. We're going to focus more on sections 4.3 to 4.8, which is the sections that deal with eukaryotic cells. Primarily, we're going to compare plant versus animal cells. In section 1, the focus is on the cell theory, which has 3 parts. The first part of the cell theory says that all organisms are composed of cells. 2 scientists are given credit for discovering or determining that first component of the cell theory. One is a botanist, and that means that he studied plants. And his name is Matthias Schleiden, and it's fine if you just remember his last name, Schleiden. Schleiden is given credit for discovering that all plants are composed of cells. And then Theodor Schwann, who is a zoologist, is given credit for discovering that all animals are composed of cells. So Schleiden and Schwann are the biologists that are given credit for discovering the first part of the cell theory, that all organisms are composed of cells. And since their evidence was discovered, since they presented their evidence, we have yet in biology to discover a living thing that hasn't been composed of at least one cell. So all of our evidence since the early 1800s has shown that all organisms are composed of at least one cell. The second part of the cell theory says that cells come only from pre-existing cells. And the scientist given credit for this discovery is Rudolf Virchow, and we call him, we can just learn his last name, Virchow. Notice that all of these discoveries are made in the early 1800s, and this is because this is when microscopes were invented. So it's the first time that we can actually study on the tissue level and the cell level. And the third component of the cell theory is that cells are the basic units of structure and function in organisms. This is an image that shows you a micrograph of leaf tissue and a micrograph of the tissue that lines the trachea of this rabbit, which is, that's going to look really, really similar to the same tissue in humans. Just an image just showing you that cells can range from nanometers to measuring in meters. But most of the time we use nanometers, micrometers. Micrometers are the typical size of a cell, okay? So you also need to know the reason that cells are typically so small is that a small cell has a larger ratio of surface area to volume. So the surface area of the cell is the plasma membrane. It's covered by the plasma membrane. So if this is a cell, this 4 centimeter cube here on the left, the plasma membrane would cover that outside and it would take up 96 square centimeters. The volume inside would be 64 cubic centimeters. Now, if we looked at these 64 1 centimeter cubes, these cubes each have a surface area of basically one square centimeter times 6, because they have 6 sides. So they would have a surface area of 6 square centimeters, but then when you multiply that by 64, you get 384 square centimeters of surface area that would be covering these cubes. That's a lot more surface area for these small cubes than this one big cube, even though they all take up the same amount of volume. The volume taken up by all 64 cubes is 64 cubic centimeters. So it shows you the surface area to volume ratio down here. For the smaller cubes, it's 6 to 1. And for the one large cube, it's 1.5 to 1. So even though their volume is the same, their surface area is much greater compared to that volume. So I always tell people which would take more wrapping paper, a present the size of the cube on the left, or wrapping all 64 of these smaller presents. They take up the same amount of space, but it would take a lot more wrapping paper for the 64 smaller cubes. And the wrapping paper represents the surface area. So why do we need so much surface area? Because that is how nutrients and oxygen get into the cell, is through the surface, through the plasma membrane. So the more plasma membrane or surface that we have, the more nutrients and oxygen can get in. There's a whole section here on magnification and microscopes. Let's just skip all the way to this page, which is slide 18. And it's showing us the three views. On the left, we're looking at a euglena, which is just a little protozoan that lives in pond water. We're looking at euglena under a light microscope, magnified 200 times larger than its regular size. And this is what we can see with a light microscope. And then with a transmission electron microscope, we can see the organelles in detail inside of this euglena, magnified 12,500 times larger than what its actual size is. And then we can look with a scanning electron microscope at detail of the surface of the euglena. Look at that detail showing the flagella, you can even see little ridges in the surface of the euglena. And it's only magnified 1,520 times larger, but it's just such good detail on the surface. So this is just letting you know the tools that we have for viewing cells today. Back in the early 1800s, the tools were simply the light microscope, but now we have electron microscopes that allow us to see details that so that we can study these cells. And what you're looking at here are different phase contrast, using the light microscope, but using different types of contrast. Contrast can come from filters, it can come from staining, or a combination of both. But phase contrast can help us see different views of the same structure. What we're looking at here are cheek cells. What's visible in all of these is the plasma membrane and then the nucleus. You can see those in all of these, but you just have like the nucleus is highlighted here so well in this picture on the right. So you just have different options with phase contrast as well. Now, moving on to section 2, we're going to briefly cover prokaryotic cells. Remember that prokaryotic cells lack a membrane-bound nucleus. So the basic difference between prokaryotes and eukaryotes is prokaryotic cells don't have a nucleus and eukaryotic cells do. Prokaryotic cells are also much smaller than eukaryotic cells. And we have 3 domains of living things. All living things are grouped into one of the 3 domains. All of the living things that consist of prokaryotic cells are either in the bacteria or the archaea. The bacteria can cause disease, but they also can be beneficial as decomposers, and they also live symbiotically inside of and on the surface of other living things like humans, and are very, very useful. So bacteria can be harmful and helpful. They can also be useful in manufacturing products and drugs. Archaea are also prokaryotes, but they live in extreme habitats, and there's so many differences between the two that they cannot be grouped in the same domain. So that's why they are divided into two different domains. As far as the basic structure of prokaryotes, they can have one of three basic shapes. We'll look at the pictures. They can either be rod-shaped, like this bacillus. They can be spiral-shaped, and spiral-shaped can either be called spirillum or spirochete. The spirochetes tend to be modal. They tend to be able to move. And they can also be round or coccus. Then if you look at the structure of a typical bacterial cell, this one is showing a lot of different surface structures that the bacteria has for protection. So bacteria, prokaryotic cells, which can be bacteria or archaea, are always going to have a plasma membrane. They're always going to have DNA in the form of a long circular chromosome that's so large that it's kind of bundled up here, but if we stretched it out, it would be one large circle. So a large circular chromosome, a plasma membrane, ribosomes for making proteins. Of course, inside the cell is still called the cytoplasm. Then we're going to have structures outside the plasma membrane, the cell wall, the capsule outside the cell wall, the structures called fimbriae, which are little hair-like bristles that allow the bacteria to adhere or stick to surfaces. The bacteria or prokaryotic cells can also have flagellum, which would help them to move. So we're looking at an individual E. coli cell here as a representation of a prokaryotic cell. But basically, prokaryotic cells aren't going to have a nucleus. They do have DNA, but it's not enclosed by a nucleus. And they're not going to have organelles like mitochondria and chloroplast and the Golgi and the ER. Now, I highlighted plasmids here because we didn't talk about that yet. And there's not a plasmid in the picture, but sometimes prokaryotic cells will have small accessory rings of DNA, small rings of DNA that are extra to the large single circular chromosome. And sometimes they'll carry extra genes like antibiotic resistance. So those are called plasmids. Now we're going to move into section 3 on eukaryotic cells, and we're going to focus on plant and animal cells and learning the names and the functions of the organelles that compose eukaryotic cells. So eukaryotic cells contain a membrane-bound nucleus that houses the DNA and specialized organelles. We're going to skip the origin of eukaryotic cells, not because it's not important, but just for time. But you can go back and look at the theory about the origin of eukaryotic cells is called the endosymbiotic theory or endosymbiont theory. You can read about that if you would like. What we're looking at here is an image of a typical animal cell. Here's a typical plant cell. These are the same images that are in your lab manual. We will focus more on learning how to label plant and animal cells, learning the names of the structures and what they look like. We'll focus more on that in lab. But I will tell you that different cells are going to be specialized and have different amounts of these organelles depending on their function. For example, liver cells, one of the functions of a liver cell is to detoxify drugs. So liver cells will have more smooth endoplasmic reticulum than other cells will. And nerve cells' function is to carry electrical impulses. So they're going to have these long branching structures called axons, and those axons are covered by plasma membrane. So a nerve cell will have more plasma membrane than other cells will.